Okay, in this module I will discuss the biological cells. I will introduce the biological cells as well as tissues. Now, this part of this course and in particular this module will only touch upon some of the key terms as well as also discuss some of the salient aspects of the cell structure without getting into significant details like what a biologist otherwise would cover. So, these are the bulleted points that I am going to touch upon one by one after other. So, we will first define what is cell. So, biological cell is defined as a self-contained unit capable of replicating itself given the proper nutrients and environments. So, let me explain some of these terms or phrases that I have used in this particular key definition. So, this one is this particular term like self-contained and so on. So, self-contained means it has all the organelles which can be used to which can perform certain specific functionalities. For example, ribosomes in cells can synthesize proteins. Mitochondria is used as a powerhouse. I will go through that next slide. So, then I will explain it more, and it is also capable of replicating itself. That means, it can propagate from 1 to 2, 2 to 4, 4 to 8, like cells can undergo cell division. And if the cell division is there, if cell division is possible only when proper cellular microenvironment exists. That means, the cell is provided with proper nutrients and environment. So, in general through nucleatic in general the cells can be classified as two classes. One is the eukaryotic, EU means truly. So, eukaryotic means truly nucleated cells which are enclosed by lipid bilayer and also contain the necessary genetic material. So, essentially that genetic material includes the nucleic acids like DNA, RNA needed to direct the continued propagation of the cell. So, there are certain things which is kind of implicitly mentioned here, like, and I will describe it in much more details in later part of this course, like nutrients and environment. So, nutrients are essentially required for the survivability or cell survival. Environment also cellular microenvironment means, so essentially this cellular microenvironment means like cell must receive certain signaling molecules, or for each cell to survive it must receive a combination of signaling molecules, and in the absence of those signaling molecules the cells cannot survive. Now, what is cell signaling mechanisms and what are the signaling molecules which are essentially proteins, those things I will discuss it later. So, now if we move to the next slide, what I was telling you some 2-3 minutes ago, and let me explain with some little bit more specific details in these slides. So, this kind of horseshoe type of structure, you see this is mitochondria. So, this is known as the powerhouse of the cells. Now, you can also see here certain ribosomes, and this is the ribosomes in the cytosol. These ribosomes essentially are the organelles which are used for synthesizing proteins. Now, other things very important is that if you see that very regular type of cell membrane structure, now although it is shown so simplistic manner in this particular cross section slides of the animal cell, but in reality the cell membrane is very important, and in eukaryotic cells it is a double layered structure Also individual organelles, as I am marking here, like a mitochondria, then you have this Golgi body as well as other organelles, they also have a very well defined double layered structure. So, this double layer membrane structure is kind of very characteristic of different organelles, of different cellular organelles. Okay. And the other things that you can see rather quite clearly. So, this is the almost like a central region, this is the nucleus. Now, nucleus contains DNA, that is D-ribonucleic acids, and also nucleus is also separated by separated from rest of the part of the cytoplasm by very well defined double layer structure, and this nuclear membrane also has certain pore structure, and these pores essentially form that nuclear pore complex. So, later on I will explain to you that in an adult healthy cells, or as a matter of fact in all the cells, the DNA can undergo transformation to RNA, which is known as the transcription process. Then RNA can go to synthesize to proteins, and this is called translation process. So, typically transcription process takes place in the nucleus, translation process takes place in the cytoplasm of an eukaryotic cell. The story is little different in case of prokaryotic cells, which is like more primitive cells and unicellular cellular cells. The other thing is important, as it is shown in this particular slide. You see that these are like yellow kind of fibrous structure. So, this is what is known as the ECM, extracellular matrix. Now, extracellular matrix contains collagen, elastin, and various other biological micromolecules, and this extracellular matrix plays certain key roles in determining the cell functionality, like the way cell would migrate, or cell motility will take place on an extracellular matrix or on a biomaterial substrate. The other things should draw your attention is that this is that cytoplasm of the cells, and you can see the cytoplasm has certain fibrous structure. So, there are three components of the cytoplasm, and this fibrous structure, although shown more clearly towards the end of this particular cross section image, but you have to believe me that this particular cytoskeleton structure extends very neatly throughout the entire cross section of the cells, and the cytoskeleton structure has three constituents. One is actin filament, then one is a microtubule, and third one is that intermediate filament. So, I repeat, cytoskeleton in a eukaryotic cell has actin filament, intermediate filament, as well as microtubules. Now, these three different components of the cytoskeleton, they have different range of their mechanical properties in terms of their elastic stiffness or deformation behavior under stress. Why I am emphasizing so much on the cytoskeleton structure? Because cytoskeleton also supports or provides mechanical support to other cellular organelles. So, from that point of view, the understanding of the cytoskeleton structure is quite important. So, just to give you some difference between the prokaryotic, that is pro means before and karyon means nucleus, prokaryotic cells and eukaryotic cells. Now, prokaryotic cells certainly are not very well developed cellular structure, whereas eukaryotic cells represent the most matured and more well developed cellular structure. In prokaryotic cells, there is no well defined nucleus like what you have in an eukaryotic cell. Instead, DNA, as you can see in the very coiled form, is distributed or dispersed in the cytoplasm of a prokaryotic cell. So, this is that major difference between prokaryotic cells and eukaryotic cells. Other difference is that that doubling time of prokaryotes is around 30 to 40 minutes. That means the prokaryotes typically divide much faster than the eukaryotes, which are like a truly nucleated cells. And for eukaryotic cells like truly nucleated cells, doubling time is somewhere around 12 hours or longer. The other thing you might have noticed in the last slide, I think there is that micron bar scale bar is not there. Otherwise, the size of the eukaryotes is much larger, around 25 to 30 micron than prokaryotes, that is 2 to 3 micron. The first two differences I have already explained to you a few minutes back. Now, coming to the cell size, as I said that cells are typically 20 to 30 micron in size, and in natural tissues like in an adult human, the cell density is typically 1 to 3 billion cells per milliliter. Now, if you consider a normal healthy person weighing 50 kg, therefore, we will have around 50,000 milliliter of and then therefore, that human body contains somewhere around 10 to the power 14 number of cells. Now, these numbers, as you see that it also depends on the body weight of a human of a person, but roughly this order of magnitude of these numbers are important, so that you have an idea that how many number of cells typically are present in human body. Other things one has to also consider that depending on the size of this, whether it is a eukaryotic cell or prokaryotic cell, you have to see that what is the total number of protein molecules, which are proteins which are present in the eukaryotic cells. Now, if you go by these very simple calculations which are mentioned in this slide, what I want to draw your attention to these numbers here. So, typically eukaryotic cell has a protein molecules is 4 into 10 to the power 9 molecules, whereas in a prokaryotic cell this protein molecule number of proteins is little less, simply because these prokaryotic cells have a much lesser cell size compared to eukaryotic cells. So, I repeat, you have a 10 to the power 14 number of cells, typically eukaryotic cells, and in terms of proteins you have a 10 to the power 9 proteins in eukaryotic cells, whereas around 10 to the power 6 proteins in prokaryotic cells. So, this some idea about these numbers or order of magnitude of these numbers is important, so that one can rationalize that once a cell is placed in a culture medium with proteins and so on. So, this how these proteins play an important role or mediate the cell material interaction. This is another fantastic image of a animal cell cross section, where each of these organelles are essentially stained with different fluorescent dyes, and as a result you can see that rough endoplasmic reticulum is stained in more or less green, Golgi apparatus appears is red, mitochondria also you can stain it by microtracker, that is one of the dyes. Typically the nucleus it is stained by DAPI stain, and actin filaments also stained with a different kind of dyes. So, essentially what this microscopic image tells you, that it is indeed possible to carefully stain each of these specific organelles using certain fluorescent or certain specific fluorescent dyes, so that one can image different organelles appropriately under a microscope. So, this has been brief discussion has been summarized in this slide. So, as I said that all the organelles in a eukaryotic cell can be stained with specific fluorochromes, for example, nucleus by DAPI, actin filament by phalloidin, and mitochondria by microtrack. And various structural features can therefore be imaged, can thereafter be imaged by fluorescence or confocal microscope. Scanning electron microscope, that is the abbreviated as SEM. This can be used to see just the cell morphology or cell spreading, but however finer details of the cellular ultrastructure can only be imaged when the cellular organelles are tagged using specific fluorochromes, and therefore to appropriately image the cell structure, one has to use different kind of microscope, that is fluorescence microscope or confocal microscope. Another microscope which is used not that widely or not that extensively, that is the transmission electron microscope. It can be used to image some of the ultrastructure like ribosome structure or DNA structure, but which requires very tedious sample preparation and very careful sample preparation, and therefore, many researchers they regularly use fluorescence or confocal microscope in conventional biological sciences as well as in the field of biomaterials. Around 5 to 10 minutes back, I was explaining you that how this cytoskeleton structure they appear in a eukaryotic cell. Now, here this particular slide shows some of the details, and essentially you have this microtubule which has a different diameter than the actin filaments, which is shown here as a red, and also this another one. So, you have a intermediate filaments also shown is with a different colors like black colors. So, microtubules, intermediate filaments as well as the actin filaments, they essentially represent different protein complexes, and also they have different morphology as well as sizes. So, from there one can distinguish in a carefully imaged cellular structure that how these three important elements of a cytoskeleton would appear in real life. I was also explaining sometimes back that although it appears very simple as a cell membrane has a very characteristic structure, what we call is a phospholipid bilayer structure. So, in this phospholipid bilayer structure you have certain transmembrane protein also. So, transmembrane protein means the protein which extends from intracellular region to extracellular region, like which extends from cytoplasm of a cell to outside space of the cell. These are called transmembrane proteins. You also have some of the specific proteins which are attached to the cell membrane. One of them has been shown here, glycoprotein, which is protein with carbohydrate attached, and another one is called glycolipid. So, lipid with carbohydrate attached. You also have a cholesterol, and all this is just in the inside of the cells, and you have a peripheral membrane proteins. So, it is like a very nice two bilayer structure. It is a kind of very sandwich type of structure, and there are several transmembrane proteins are also shown, and also you have a certain channels in this cell membrane structure. Now, these channels are very specific channels, and many researchers in a biological sciences department they do extensive research on the characteristics and properties of these channels. So, one of these channels can be sodium potassium channel. Sodium potassium channel essentially has some specificity that it can allow only sodium intake or exit, or only the potassium intake or exit. At the same way, cell membrane also have a voltage gated calcium channel. So, if these channels essentially will sense certain voltage differences across a membrane, and accordingly they will open up only to allow the transport of the calcium ions. I repeat, there are two type of channels on the cell membrane. One is called sodium potassium channel, another one is called calcium channels. Now, these calcium channels, they are voltage gated calcium channels. Now, these channels are extremely sensitive to this voltage difference. So, at a particular voltage difference, these channels will allow the specific transport of only calcium ions. Similarly, some channels are there in the cell membrane which are shown differently in terms of their morphology, particularly these type of channels. So, these channels are essentially can be sodium potassium channel. Again, they can specifically allow the transport of the sodium or potassium ions through this channel into cell or outside the cell. Here I must mention that any given time point there is a kind of a balance in terms of the sodium, potassium, calcium ions between the intracellular region and extracellular region. And the difference in terms of this ion concentration between the intracellular region and extracellular region causes certain transmembrane potential across this cell membrane, and this transmembrane potential is of the order of 70 millivolt or so on. But you have to also understand that this thickness of this cell membrane is very small. It is like few nanometers, like 2 to 3 nanometers or something like that. Now, although this transmembrane potential is very small, 70 millivolt, but if you consider the voltage difference across the cell membrane, it is 70 millivolt divided by 2 to 3 nanometer. Then nanometer means 10 to the power minus 9 meter, millivolt means 10 to the power minus 3 volt. So, therefore, you can immediately see that it is kind of turns out to be 35 into 10 to the power 6 volt per meter, that is a roughly that is that transmembrane potential or voltage difference. So, it is essentially this value represents very significant voltage difference across the cell membrane simply because of the difference in the ionic concentration in the extracellular and intracellular region. Okay, this is what has been mentioned to you just few minutes ago, that importance of the cell membrane structure. You have sodium ions, you have a potassium ions, and the sodium ions can be transported through some of the specific channels. Potassium ions can be transported through these channels. This specific pore arrangement at cell membrane allows the diffusion of sodium, potassium and calcium ions, and the cell surface receptor proteins are there, and they play an important role in mediating interaction with a biomaterial substrate. So, other specific characteristics of a nuclear membrane, as I mentioned in the beginning of this module. So, nuclear membrane also has a characteristic bilayer structure. You see, this is the inner layer and this is the outer layer, and this inner layer and outer layer, they have a very specific arrangement, and the way you see that there are certain holes in that, the way I am drawing, these are certain holes in the nuclear membrane structure, and these holes they essentially form what they call nuclear pore complex. So, this nuclear pore complex is one of the things that also allows the transport of the proteins or certain cellular signaling molecules into the nucleus from the cytoplasm. So, as you can see from last 15-20 minutes or so, that cells have the unique cellular organelle characteristics which also allows the transport of various ions, proteins and biological macromolecules. So, this gives little bit more details about what I have shown you in this particular slide, that what is the subcellular structure of the eukaryotic cell, or in other words what is the cytoskeletal structure of the eukaryotic cell, because skeleton means like a human skeleton, you know that human skeleton is, so cellular skeleton essentially means that is the skeletal structure which supports, which provides mechanical support to all the cellular organelles in the cytoplasm. Therefore, one is to understand that what is the key characteristics of this kind of cytoskeleton structure that makes it so special in terms of providing mechanical support. Now, you can see that I will start with the bottom one, that is the actin filament. As I said that this essentially represents the protein complexes or polymeric long chain, and here these polymeric chains are arranged in a specific manner along the backbone chain, and these actin filaments during the cell fate processes they can undergo depolymerization. Depolymerization essentially means that these actin filaments can break down to individual monomeric molecules, now individual monomer. Now, if it is required, these monomers can come back together to make again actin filamentous molecule, and this is called repolymerization. So, the sequence of this depolymerization and repolymerization take place in a cell in a cellular structure. The second structural element of cytoskeleton is intermediate filament, As you can see from the schematic of these two microtubule, as you can see from the schematic of these two microtubule and actin filament, microtubule has a much larger or relatively larger dimension in terms of the tubule diameter compared to the actin filament. Third one is that intermediate filament. Intermediate filament appears to have a intermediate size between the actin and the microtubule. Now, this has been shown very nicely in this part of the cell membrane structure, where you can see this is your mitochondria and this is the different filamentous structure in the cytoskeleton, which is very nicely being shown, and this is your intermediate filament, and some of the microtubules are shown here in the form of a large tubular structure, and also you have a actin filamentous structure which are shown here. So, you are seeing this green color, slightly green, light green color, this is your actin filaments. You see dark green colored large tubular structure, this is your microtubule, and you also see that intermediate filaments with a characteristically different morphology compared to the other two filamentous structure. I think I will stop now and I will come back to this more discussion on the cell structure.